Today we will talk about the gene mutation. What this gene mutation is? Gene mutation is simply that there is an alteration or change in the base sequence of the DNA. We know that कि DNA जो है इसमें nitrogenous bases होती है और अगर कभी कभार ये नाइट्रोजन बेसिस इनकी सीक्वेंस चेंज हो जाएगी तो हम इसको म्यूटेशन बोलेंगे. सो व्हाट दिस म्यूटेशन इज? म्यूटेशन, जीन म्यूटेशन इज सिंपली और sometimes called as DNA mutation. Gene mutation is simply alteration or change in the base sequence of DNA. The base sequence of DNA. Gene mutation is simply the alteration or change in the base sequence of the DNA. Or any gene can mutate. However, mutability differs from gene to gene. और जब म्यूटेशन होती है जीन की तो दिस जीन कैन sometimes it can revert back to the wild type. और this mutation जो है gene mutation, it mostly occurs in the recessive genes. So mostly gene mutation recessive genes में होते हैं. It occurs in recessive genes. However, very few mutations occur in dominant ones. This gene mutation जो है, it can be gene mutation. It can be lethal भी हो सकते हैं. It can be neutral. इसका कोई असर नहीं हो सकेगा. And then it can be advantageous. And sometimes जो जीन म्यूटेशन जो है it results in the loss of the function of the gene. जब एक जीन में म्यूटेशन हो जाएगी तो इसकी जो ओवरऑल फंक्शन जो है वो that will get lost, वो पूरी खत्म हो जाएगी. And this sometimes it can be neutral. This mutation would have no effect on the protein, on the synthesis of the protein. Then we have this mutation. It was first recorded by Hugo de Vries. And he recorded it in an ancon sheep. It is a short-legged sheep. कि इन्होंने देखा कि एक ग्रुप ऑफ शीप्स है, उनमें से एक शीप जो है उसके जो दैट लेग्स आर वेरी शॉर्ट, तो इन्होंने कहा कि ये जो है इट इज़ बेसिकली जीन म्यूटेशन. एंड कॉन शीप, इट इज़ अ शॉर्ट लेगड शीप एज कम्पेयर टू द अदर शीप्स विच हैव लॉन्ग लेग्स. देन दिस जीन म्यूटेशन, it occurs by means of a substance that is called as mutagen. So what is a mutagen? Mutagen is any substance or agent that induces mutation. और gene mutation जो है, the substance or the agent which causes mutation in a gene that induces mutation, that is called as mutagen. It can be a chemical, it can be radiation, or it can be a base analog. Then this gene mutation, it is also called as point mutation. Gene mutation is also known as point mutation because कि कभी खबर in most of the cases single nitrogenous base change हो जाती है by means of the mutation. That is why we call a gene mutation as a point mutation. Gene mutation is also known as point mutation because in most of the cases the single nitrogenous base gets altered, it gets changed by means of the mutation, by means of gene mutation. So gene mutation is also known as point mutation. और कभी कभार gene mutation can be it may involve large segment of the DNA. Sometimes it may involve many nucleotides, and sometimes it can be as small as a single nucleotide will get altered. कभी कभार एक nucleotide alter हो सकता है. तो for example we have A T A A C C T. This is a DNA sequence of nitrogen bases on DNA, and only one nucleotide will get altered. For example, this will get altered. So it can be as small as a single nucleotide will get altered or will get changed. और in most of the cases ये देखा गया है कि सिर्फ एक ही नाइट्रोजन बेस चेंज हो जाते हैं, जिसकी वजह से हम जीन म्यूटेशन को पॉइंट म्यूटेशन का नाम देते हैं. और जीन म्यूटेशन जो है, इट इज ऑल्सो कॉल्ड एज कॉपी एरर म्यूटेशन. कॉपी एरर म्यूटेशन. It has been found that gene mutation, this mutation of DNA usually occurs during the replication of DNA. So we call it copy error mutation. Why gene mutation is called as copy error mutation? Because most of the alterations in the base sequence of DNA occurs during the replication process. So it is called as copy error mutation. Most mutations occur during replication. So gene mutation is also known as copy error mutation. So gene mutation also known as copy error mutation. Then we have three methods of gene mutation. Methods of gene mutation. We have three methods of gene mutation. One is inversion. Then एक method है gene mutation का, that is inversion. Then we have substitution. Then we have frame shift mutation. Frame shift. We have three methods of the gene mutation. One is inversion, substitution and frame shift. अब ये substitution जो है, it is of two types. One is called as transition or transversion. और frame shift जो है, it is of two types. One is called as insertion and deletion. So we have to discuss these one by one. What is inversion, what is substitution and what is frame shift. First, what is inversion? This inversion, it is a type of mutation where the base sequence of DNA gets altered in a reverse order. That means here base sequence of DNA gets altered in a reverse order. For example, when this inversion takes place, the base sequence of new DNA would be entirely different from the original sequence. That means we have original sequence. This is original sequence. Original base sequence. We have original base sequence. For example, we have A T C A G A T C A A. This is the original base sequence. Now there would be inversion. Inversion. What will happen? the base sequence of DNA will get reversed. So we have, this is A, T, C, A here, G, A, T, C, A. This is now new base sequence. We have A, T, C, A. We have this, then this T will be here. This T is here, A is here, and G is here. C, then A and A. It means that the sequence was A, T, C, A, G, A, T. Now because of the inversion, this has inverted. This DNA segment has inverted. As a result of this T is now here, this is A here, and G has come to this position. So we can say that the base sequence of DNA has changed in a reverse order. So in this original sequence we have GAT. In the new base sequence we have TAG. So this is an inversion. In case of inversion, the base sequence of DNA gets altered in a reverse order. As a result of that, the original base sequence and new base sequence that are entirely different from each other. The new base sequence would be different from the original base sequence of the DNA because of the inversion. So here we have taken the example of this DNA segment. The inversion has taken place at this position. So this T will come to this location and A will come to this position. So this is the inversion has taken place. This is G is here, A and T. Now at this position T is here, A, and then instead of T we have G. So this is an example of inversion. Then we have substitution. Substitution. Substitution also known as replacement. We have two types of substitution. One is transition. Substitution simply means that nitrogenous bases will get replaced with each other. So replacement of nitrogenous bases. So in case of, if we'll talk about the nitrogenous bases, we have DNA. Here we have two types of nitrogenous bases. One is purinous. We have purinous and we have pyrimidinous. We have purinous, we have pyrimidinous. Purine is adenine and guanine. Pyrimidine is thymine and cytosine. So in a DNA we have two types of nitrogen bases, purines and pyrimidines. Then there are two types of adenine and guanine are purines, and thymine and cytosine are pyrimidines. So together we have four types of nitrogen bases: adenine, guanine, thymine and cytosine. So there is a replacement of these nitrogenous bases. So in case of substitution, there is a replacement of nitrogenous bases, and it is of two types: transition. In case of transition, one nitrogen base is replaced by another nitrogen base of its own type. One nitrogenous base is replaced by the nitrogenous base of its own type. That means in case of, if purine will be replaced by purine and pyrimidine will be replaced by pyrimidine. So this is what is called as, in case of transition, one purine is replaced by another purine, and in case of pyrimidine is replaced by another pyrimidine. For example, if we have purine A, it will be replaced by G or vice versa. We have pyrimidine T, thymine, it will be replaced by cytosine and vice versa. For example, we have examples for the first one. Purine is replaced by purine. We have base sequence A, TT. Here A is a purine, it will be replaced by purine. So A is replaced by G. A is replaced by G. So here we will have GTT. So this is when the purine, adenine is a purine, it will be replaced by another purine, that is G. So new base sequence is GTT. The original base sequence is ATT. Then we have pyrimidine. For example, we have pyrimidine TAA. T is replaced by C. We know that T is pyrimidine, and this will be replaced by cytosine. So CAA. This is original base sequence, this is new base sequence. Here pyrimidine, thymine is a pyrimidine, it is being replaced by another pyrimidine. So we can say that in case of substitution there is a replacement of nitrogenous bases. अब replacement of nitrogen दो types की होती है, transition और transversion. अगर similar types इसमें replace होते हैं, for example purine replace होती है by means of purine nitrogen base, और in case of transition, purine is replaced by another purine, and pyrimidine is replaced by another pyrimidine. This is what is called as transition. That means one nitrogen base is replaced by another nitrogen base of its own type. Purine is replaced by purine, and pyrimidine is replaced by pyrimidine. This is what is called as transition. Then we have got another transversion. In case of, we have transversion. In case of transversion, purine is replaced by pyrimidine. Purine is replaced by pyrimidine. Purine जो है वो replace हो जाएगी by means of pyrimidine. That means we have, for example, A is A is replaced by T, or A is replaced by C. Same is the case, G is replaced by T or C. So we can say that we have here purine, it is replaced by pyrimidine in case of transversion. In case of ट्रांसवर्शन एक प्यूरिन जो है उसको रिप्लेस किया जाता है एक पैरामिडिन से. दैट मीन्स वन प्यूरिन इज रिप्लेस्ड बाय पैरामिडिन इन केस ऑफ ट्रांसवर्शन. सो विल टेक एन एग्जांपल. फॉर एग्जांपल वी हैव AGG. We have AGG. This A is replaced by A is a purine. It is replaced by A is purine. It is replaced by T. So it would be TGG. Same is the case. We have can be also replaced by C. So AGG. This is original base sequence. Here A is replaced by C. So we have CGG. Same is the case. We have, for example, we have GAA. This G is replaced by C. G replaced by T or C. So we have CAA. Then G is replaced by guanine is replaced by thymine. That is purine is replaced by pyrimidine. So we have TAA. So this is mutation, because the original base sequence is entirely different from the new base sequence. So this was regarding the gene mutation. In the next lecture we will discuss about the frame shift mutation. So today we will talk about the gene mutation. Gene mutation is simply the alteration in the base sequence of the DNA, and sometimes in most of the cases this gene mutation, it involves a single nitrogenous base. Therefore gene mutation is also known as point mutation. And most of the gene mutations occur during replication, so it is called as copy error mutation. These mutations can be lethal, they can be advantageous, they can be sometimes neutral. And in most of the cases gene mutations, they occur in recessive genes and very few occur in dominant ones. When a gene undergoes mutation, there is possibility that it can revert back to the wild type. It can revert back to the wild type. And these mutations are of three types. We have inversion, we have substitution, and we have a frame shift. So in case of inversion, when inversion occurs, there is the base sequence of DNA gets changed in a reverse order. So we have discussed that. Then we have substitution. There is a replacement of nitrogenous bases, and it is of two types. One is transition and transversion. Transition, when purine is replaced by purine and pyrimidine is replaced by pyrimidine, this is simply a transition. Then transversion, when a purine is replaced by pyrimidine, so that is transversion. In the next lecture we will discuss about the frame shift mutation. Thanks.